is abnormal rhythms. All of you know that if you feel your pulse, it's nice and rhythmic. It should be, especially for young folks like yourself. But many of you in here will know somebody who develops what are called premature atrial contractions, so-called PACs. They may develop so-called premature ventricular contractions, PVCs. They may suffer from atrial fibrillation. About 2 million people a year develop atrial fibrillation. Not a death sentence by any stretch of the imagination, but not a fun thing to have happen either. And then there's ventricular fibrillation. And that is an emergent condition. Somebody goes into VFib, ventricular fibrillation, unless they spontaneously convert on their own, which is pretty rare, unless you have a defibrillator, they'll be gone. So ventricular fibrillation is a terminal event. Anyways, we're going to talk about that. Then that will take us into heart sounds, which will then take us into the lab for tonight. As part of talking about heart sounds, I'm also going to basically introduce you to heart murmurs. A murmur is just an abnormal heart sound. That's all it is. And that's due to the valves either not closing and forming tight seals or through other phenomena as well. So those are the objectives, and then we'll get into the lab tonight, where you'll actually record both the EKG and the heart sounds of your subject in your group. And that's what we're going to do. That's the focus for tonight. So given that, let's get the show on the road. I know many of you in here study this material on a regular basis, but if that's not you, then I would encourage you to start studying ASAP, because there's a lot of information on this text. I could be wrong, but that's just my impression. That's the way that I would encourage you, all of you, to prepare for this next test. All right, so what we're going to do is I'm going to show normal EKG up on that top line. Then I'm going to present something to you that will be a little bit different from normal. And I'll also do something on the bottom that will be different from normal as well. So in talking about EKGs, you should see a P wave that precedes the QRS complex. All of you should know that the P wave represents atrial depolarization. That's the electrophysiological event. The QRS complex represents ventricular depolarization. The T wave represents ventricular repolarization. And this process repeats itself over and over and over again. So here's a P wave. Then we got a Q, R, S, T wave, and this repeats itself over and over and over again. That's my stockbroker calling. All right, let's see how well I did with this. Not too bad. first thing you're going to do when you look at an EKG is you're going to look at what's called the R to R interval. I didn't do it perfectly, but it's not too bad. So the R to R interval is simply this interval from one R wave to the other. And hopefully they look pretty uniform. What do you guys think? If you look at the R to R intervals, they look fairly similar to one another. Yeah, no, maybe so. Approximately. So right out the bat, what that's telling you is that there's this nice rhythmical set of events, electrical events within the heart. The next thing is that we're going to look for the presence of P waves. All of you should know that the SA node, the sinoatrial node, is the pacemaker of the heart. And the P wave represents atrial depolarization. So we're always looking for the presence of P waves in front of the QRS complex. We see that here so that we know from this information that the conduction pathway is beginning at the SA node, which is in the right atrium, and then is spreading down through the remainder of the conduction pathway in the heart, ultimately to the ventricles. That's represented by our QRS complex, and then the T wave. And if we take a look at the anatomy of the heart, so up top, meaning here, we've got the individual atrium, and then below we've got both the right and left ventricles. I'm talking about the conduction pathway of the heart. There's a cluster of cells within the right atrium. I'm just going to put an asterisk there, and this is the SA node. That's where every normal heartbeat is initiated. And at the SA node, this is referred to as the pacemaker of the heart. And that word pacemaker is a very important it's a very important pacemaker. It's a very important word. Here's a graph. On the y-axis, we've got membrane potential in millivolts. Start out at minus 100 millivolts and go up in increments of 50 millivolts. We've got threshold. We'll say the threshold is at 40 millivolts. Excuse me, minus 40 millivolts. You guys all remember this, right? Sort of, possibly. Here's the resting membrane potential, we'll say, of a neuron. resting membrane potential at minus 70 millivolts. That was our textbook value. But here's the thing that's cool about these pacemaker cells. A key property. Is that they have a unstable resting membrane potential. What does that mean? That means unlike a neuron, which would have a constant resting membrane potential, for pacemaker cells, here's what they do, is that they come along and they spontaneously move towards threshold. And when they reach threshold, all of you would tell me they produce an action potential. Then they repolarize. And then they start drifting up towards threshold again. And when they hit that, they produce an action potential. And this process just repeats itself over and over and over again throughout the course of our lifetimes. So, pacemaker cells have unstable resting membrane potentials. In other words, they spontaneously rise towards threshold. And when they reach threshold, then they produce an action potential. This is an inherent property of all pacemaker cells. So these cells here in the SA node are pacemaker cells. They determine, they dictate the heart rate. Now, all of you know that your heart rate can vary. Sometimes it can slow down, sometimes it can speed up. Well, sit there and think about this. Is that how could we cause a pacemaker cell to fire at a slower rate? We change the rate at which it depolarizes. So where we had 2 events before, now we only have 1 event. And if you sit there and think about exercise, with exercise, all of you know that your heart rate goes up. The pacemaker cells move towards threshold at a faster rate. So during exercise, you would see something where the slope of moving towards threshold is much faster. And that's pretty cool biology. So for instance, these neurotransmitters that are being released by the sympathetic neurons, they're affecting the rate at which we move towards threshold. I forget it. Can anybody help me out here? Parasympathetic neurons release what type of neurotransmitter? Andrew, you remember?
No, I'm a little rusty.
You're a little rusty on that, but you're gonna really polish off that rust real quick. Miss Knudsen, how about you? Can you help me out? What's the chemical message or release from parasympathetic neurons?
It's not acetylcholine.
Say louder.
It's not acetylcholine.
It's not acetylcholine.
It's
Miss Lake.
yeah.
Kathy Lake. What would you say? Miss Chan. She said norepinephrine. Here's my response to it. Beep, beep, beep. Oops, that wasn't the right answer. Miss Elizabeth. patient,
Oh,
Mrs.
wait,
Jones,
it is
is in
acetylcholine.
deep SHI.
It
It is acetylcholine.
There we go.
But you said it's not
It's not.
acetylcholine. It is acetylcholine.
It is. Norepinephrine is what gets you going. Yeah.
Folks, write this in your notes. The parasympathetic nervous system has a positive or negative chronotropic effect. Is all.
I said negative.
That's correct. So ACH changes the slope at which we move towards threshold, so it's going up towards threshold at a slower rate, which means heart rate will go down. Sympathetic stimulation causes heart rate to go up. And how does it do that? Because the norepinephrine, through its activity on adrenergic receptors, causes the cell to depolarize at a faster rate. That's pretty cool. All right, so we've got our pacemaker cells and the SA node fired. That then leads to depolarization of the atrium. So I'm just going to put number 1 by the SA node. We're going to put the number 2 by these green arcs. This is the depolarization of atrial cardiac muscle. There's another cluster of pacemaker cells. And these pacemaker cells are found between the atrium and the ventricle. So this is the AV node, the atrioventricular node. The AV node. And so at the AV node, we've got pacemaker cells. The inherent rate for the SA node, the inherent rate of these pacemaker cells is 60 times a minute. I should say intrinsic rate. That means I could come along and cut the vagus nerve. And all of you tell me, if I cut the vagus nerve, the heart's going to have a heart rate of, say it, Austin, 60 beats per minute. That's exactly right. That's the intrinsic property of the SA node. The intrinsic rate of the AV node is 40, we're going to say about 40 beats per minute. And let me just modify that. We're going to say about 45 beats per minute. Simple question. If we've got 2 groups of pacemaker cells, why is it that the SA node is the pacemaker of the heart? Why isn't the AV node the pacemaker of the heart? And the answer is because the pacemaker cells of the SA node are getting the threshold much quicker than the pacemaker cells of the AV node. And therefore, it's the SA node that dictates the heart rate under normal conditions. Now, the The SA node and the AV node are connected by a conduction system. I'm putting this in purple. And this is known as the internodal pathway. Inter meaning between. Internodal pathway. So that's number 3. The depolarization of the internodal pathway. Number 4 is the depolarization of the AV node. Coming off the AV node, there is what is known as the bundle of His. So collectively, these conduction pathways are known as the bundle of His. And there's a so-called right bundle branch, a right bundle branch. Here I'm going to put an arrow pointing here. I'm going to put R, B, B. You're just going to say, or read, that's the right bundle branch. And there's a left bundle branch. Miss Maria, the left bundle branch is going to control which ventricle?
The right one?
Here's the question, just to make sure you're hearing.
Sorry.
The left bundle branch, left, right, left, right, left, right. The left bundle branch is controlling which ventricle?
The left ventricle.
Fantastic. And you would tell me that the right bundle branch is controlling?
The right ventricle.
That's correct. So we're going to say, number 1, depolarization of the SA node. Number 2, and somewhat simultaneously, number 3, we have the depolarization of the atrium, the depolarization of the internodal pathway. That gives rise to number 4, which is the depolarization of the AV node. That then gives rise to the depolarization of the bundle of His. And then there's like this specialized conduction pathway that provides like a network within each of the ventricles. And I'm just going to represent it like this little tree type diagram in purple. And these would be known as the Purkinje fibers. And that's number 6. And then that gives rise to number 7, which is the depolarization of the ventricular cardiac myocytes. So that's the conduction pathway within the heart. So now we're going to come back to our illustration over here on the left. And now, remember, first thing you're looking at is the RR interval. That right off the bat tells you whether there's an arrhythmia or there's a normal rhythm. If the RR intervals are normal, by definition, the rhythm of the heart is normal. But if there is a difference in the RR intervals, by definition, there is an arrhythmia. So let me show you something that would fall into that category. this works. Yeah, so in the second trace, hopefully it's very obvious to you that the R intervals are not uniform, yeah? That makes so? Well, this first R to R interval does not look normal. It's short. Yes?
You said earlier, by definition, inconsistent R intervals are attributed.
Yeah. What I said is that's an arrhythmia. We don't know what type of arrhythmia yet. That's where we're headed. Here's a P wave. Here's a QRS complex, T wave. Here's a P wave. Does that P wave look different from the other P waves? Maybe I wasn't made it obvious enough. Does that P wave have normal shape, Mr. Pat? When I look at this P wave, I just see a single waveform. When I look at this P wave, I see a single waveform. Here I see a single waveform. Single waveform, single waveform, single. Here, is there a single waveform there? There actually is like a little notch in that P wave. And that little notch tells you that the conduction is not normal. The conduction in the atrium is not normal. The conduction pathway was not normal. And this represents what's called a premature atrial contraction. The word premature should be very obvious to you. I'm just going to come along, put a little lasso around here, and I'm going to put P, A, C. So you can have a PAC, that's for the most part. If you have PACs infrequently, that's for the most part a non-event. Nothing to get concerned about. It may not feel right. You may sit there and be feeling your pulse, and it goes beep, 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 beep. And you go, oh, what was that? But for the most part, under resting conditions, your atrium are assist pumps. And they're not responsible for the full filling of the ventricles. So no harm, no foul if you're having those relatively frequent. Yeah.
So in this example, we're seeing the PAC because the R interval is short.
We see, so great question. So she's sitting there saying, we're saying this is a premature beat because we see a shorter R interval. The question is the premature beat due to something in the atrium or in the ventricle? So this is the beauty of your question, because I didn't make that clear. Here's the answer. Since there's a P wave, we know that that premature beat initiated somewhere in the atrium. Does that help? So you're going to look at those RR intervals and you go, okay, I see a shorter RR interval. Is there a P wave? If there is a P wave, by definition, that's a premature atrial contraction that was initiated within the atrium itself. Everybody clear on that? Yeah, go for it.
So the premature atrial contraction is what causes the shortened RR.
That's correct. So premature electrical activity in the atrium causing a shorter artery. Yeah.
Why there's a short,
So we have a shorter artery interval. By definition, there's some type of premature contraction. And now you're going to look at the QRS complex and you say, is there a P wave in front of it? And if the answer is yes, then you know that's a premature atrial contraction. Okay? All right, let's do a different variant on this. Okay, you take a look at that. That's a gross exaggeration. But there's something very interesting. Is that here we have a normal looking QRS complex. That's ventricular depolarization. Here we have a normal QRS complex, normal ventricular depolarization. But here we have a QRS complex that looks very bizarre and it's very wide. Do we see a P wave? Is there a P wave in front of this QRS complex? I see a P wave here, QRS, I see a T wave, but then I come over here and I look for a P wave and I don't see one. On top of that, I see a QRS complex that is wide and bizarre looking. That terminology is really important. PVC. Premature ventricular contraction. Premature ventricular contractions can be benign, meaning they're nothing to be worried about, or they can be harbingers of bad things to come. Miss Elizabeth. This RR interval, short or long? It's long. Okay, we're at problem solving time. We're into our algorithm for our cardiac output. What's going to happen to EEV? Take a look at your cardiac algorithm. I'll give all of you a minute or two to look at your cardiac output algorithm. The question is, with this event here, here's what I'm talking about. This R to R interval. what's happening to filling time. And based upon what's happening to filling time, I need you to tell me what's going on with EDV. Filling time increases, and therefore EDV is going to go up. Very good. Next question. EDV determines something else, does it not? What does it determine?
Partially ESV.
It determines ESV, right? That's your answer, which is fantastic. That's the right and brilliant answer. And so with change in preload, we're going to increase the preload on the ventricle. We're going to stretch the sarcomeres out longer. And that means those sarcomeres are going to contract with where are we in the line tension diagram? Are we in the descending limb, the plateau, or the ascending limb? The heart only operates in the ascending limb, right? And because we're filling up the heart with more blood, we have an increase in the sarcomere length, and as a result, because of the length tension relationship, the sarcomeres are going to contract with more force, the same force, or less force. More force, that's right. And so when you talk to individuals who are having premature ventricular contractions, the only way they know that they're having premature contractions is because they feel what, Mr. Cow?
Lightheadedness.
Lightheaded, okay, all right. I'm thinking something different.
Irregular heartbeat.
They feel irregular, but how do they feel that? How do they know they're having a irregular heartbeat? They have what's called a palpitation, right? That palpitation is the heart contracting harder, and it bumps up against the walls of the thoracic cavity, and you feel that, and you sit there and go, ooh, what was that? And that's referred to as a palpitation. That's what they're feeling. And that's all due to this long compensatory pause that occurs between this premature contraction and the subsequent normal contraction. It's kind of like somebody takes your heart and they slam it up against your chest wall and you feel it. Now, in some individuals, so we've got two different types of arrhythmias that I've described for you. Premature atrial contractions versus premature ventricular contractions. I've told you how to be able to discern the difference between those two. Is it possible that an abnormal EKG could show up on test number 4? Is it possible that abnormal EKG showed up on your homework assignment? So
what is
you
that?
can't have a PAC PVC.
Yeah. Is there a requirement that there's no P-way for the PVC?
The requirement for diagnosing and determining whether there's a PVC is to look at the QRS complex and ask yourself the following question: is it wide or is our And if the answer is yes, then by definition you know it's a PVC. Yeah.
So that then, the P wave is just being obscured by the large QRS complex, if not engaging.
Yeah, except what's happening is there may be no P wave because the electrical activity of the heart. So normally, the electrical activity starts here, leads to depolarization of the atrium, then we have depolarization of the AV node, depolarization of the bundle of His, depolarization of the Purkinje fibers, and that leads to these waveforms here that are very consistent from one QRS complex to the next. But when the R-to-R intervals change, or the durations of a particular wave change, by definition, the conduction through that, whatever part of the heart that is abnormal. So, coming back to your question, is that the electrical activity of the heart could be initiated by sick or injured cells in the left atrium, or sick or injured cells in the right atrium that are not the SA node. So that premature atrial contraction, I'm glad you asked this because I really want to make this clear. Is that the electrical activity of the cardiac cycle is initiated somewhere else besides the SA node. And that's because those cells have been damaged in one way or another. They become irritable, if you will. So, if we have an individual who's got a high preponderance of PACs, then and by the way, you could have premature atrial contractions that have different morphology, different structure. And that's telling you that there's more than one spot within the atrium that's initiating these cardiac events. That's telling you that the illness of your cardiac muscle is more widespread rather than being in a single spot. So you could have PACs that all look the same. That's telling you that there's a cluster of cells in the atrium that are damaged, but it's that cluster that's initiating all these premature peaks. But as soon as you see different shaped P waves that vary from one complex to the next, that's telling you that these are coming from multiple places in the left atrium. That's telling you that the sickness, the illness, the damage to the heart is more widespread just rather than it being a single cluster of cells. And the same thing is true for PVCs. You're going to have PVCs that look quite different from one another. That's telling you that there are multiple foci. Great question. Thanks for asking. Is that clear? Clear? Question: Why is the QRS complex so wide? And here's the answer: is that if the conduction is normal, then we've got the Purkinje fibers that are activating all these ventricular cardiac muscles at the same time. And so we have this nice, narrow QRS complex. But, what if we've got some sick ventricular cardiac muscle cells and they become the pacemaker? Here's the problem. Is that now, because the way the ventricle is built, it's a syncytium. It's a large collection of cells that operate as one until we have premature ventricular contractions. And now the depolarization begins with one ventricular cardiac myocyte acting as a pacemaker, and it's slowly, rather than taking advantage of this beautiful conduction system, now the conduction is much slower, going from one cardiac muscle cell to the next through this syncytium-like network. And that causes the QRS complex to become very wide and be bizarre, because the pathway in which depolarization is occurring is much slower and very abnormal. Great questions. Coming back to this strip, this is part of your homework assignment. Hopefully, based upon what I've just mentioned to you, you'll tell me what's going on here. Here I can see a P wave, I see a Q wave, a R wave, an S wave, a T wave, and then I come over here, and all of a sudden I look at this, here's my RR interval, and I see something, and you guys are going to tell me that this electrical event here is a what? Is that a premature atrial contraction or a premature ventricular contraction? And Miss Elizabeth says that's a PVC, which is exactly 100% correct. Now check this out. If we had an individual who's having multiple PVCs, but they all have this same structure, by definition, you sit there and say, that's coming from a single cluster of cells in the left ventricle that are acting as a pacemaker of the heart. But you see that here. It said, here's what I see. I see another normal P wave, a Q wave, a R wave, a S wave, a T wave. And then I come over here, and here is the QRS complex right here. You would sit there and say that that falls in the category of being wide and bizarre looking. Does it have the same morphology as that one there? Is that PVC there? No, it does not. And that's telling you that that's coming from a different part in the ventricles, different region. Let's take a look over here. What are you guys going to tell me what's happening here? I see a P wave, Q, R, S, T, and then I see something over here that's at least that wide. What is that? Is that a PAC or is that a PVC? Say it loudly.
PAC.
It's a PVC, that's correct. PVC. It's wide and bizarre looking, right? The QRS complex is wide and bizarre looking. I don't see a P wave over here. Here's my QRS complex. It's very wide. That's a premature ventricular contraction. Does that PVC look like the other PVCs? No, it does not. And that's telling you that that's coming from a different part of the left ventricle. This individual has multiple foci that are producing these PVCs, and that's telling you that there's a significant pathology here. And not only that, but they're occurring at a relatively high frequency. It's not like you can have this beep, 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 beep for the next 2 hours. Here's a normal beat, abnormal beat, normal beat, abnormal beat, normal beat, abnormal beat. This is known as bigeminy. Every 2 QRS complexes will have a PVC. I don't expect you to know any of this, but this is a high PVC load. This is something that a cardiologist, electrophysiologists are going to jump all over. Based on the EKG and the blood chemistry data, what do you think is going on? Blood chemistry data. There's some blood chemistry data right there. What is going on, Miss Jocelyn? The concentration of potassium is high. And when the concentration of potassium is high, Are we gonna be closer to threshold or further from threshold? Are we gonna be more depolarized or hyperpolarized? I guess you need to know the Nernst equation to really figure that out. Okay, last part, then we'll get going on to the lab for tonight. Any questions about that before I move on? So tonight, for your lab, you're going to come along and simultaneously record the EKG and the heart sounds. And what you're going to find is if this works right, you're going to have what's called the first heart sound and the second heart sound. Oops. I'm not going to do the right side, but we could be doing that. All right, we've got the left AV valve. What I'm going to describe for the left side applies to the right side. So, again, this is just good, simple logic. If you sit there and think about what is it that causes these heart sounds, and the simple illustration, I've used this with you guys before, is here's a heart valve, and when it opens, that doesn't make any sound. But when it closes, it's the closing of heart valves that create so-called heart sounds. So here's blood moving from the left atrium into the left ventricle. And the left ventricle fills with blood. Where am I on the pressure blind diagram? Anybody help me out? Get your pressure blind diagrams out. Where am I? Pressure blind diagram. Where are we in pressure blind diagram? Are we in phase 1, phase 2, phase 3, or phase 4? So where are we? Phase 1, that's exactly right, because in phase 1, we have ventricular filling. That's exactly right. Don't go away because I'm coming right back to you. So the blood fills the left ventricle. And now, we move into phase 2. And as soon as the ventricle is turned on, as soon as the left ventricle begins to contract, blood is pushed up against the leaflets of the left AV valve, causing the left AV valve to close. What's the first heart sound, Miss Elizabeth? That's due to what? Closing of the in this class, it's the closing of the, try it before you buy, yeah, you can say bicuspid valve. But in this class, we're using a totally different nomenclature. We're going to say the closure of the right AV valve? The left AV valve. That's correct, the left AV valve. But remember, the same thing is happening simultaneously on the right side as well. They're both in synchrony with one another. So the first heart sound represents the closure of the AV valves. That's what it represents. Well, take a look at where the first heart sound is occurring. It's occurring immediately after the QRS complex. What does the QRS complex represent? Miss Maria, what does the QRS complex represent? QRS complex, Mr. Looming, represents what? Tyler? Ventricular depolarization, right? So in order to get the ventricles to contract, we have to first get them to depolarize, and then they contract. And then they begin to increase pressure in the left ventricle, which causes the AV valves to close. But you got to understand that the first heart sound occurs immediately after the QRS complex, because that's the depolarization of ventricles. Then they create ventricular pressure, which causes the AV valves to close. Ms. Monaco, where's the second heart sound occurring?
At the aortic valve.
No. I'm talking about in reference to the EKG. Does it seem reasonable on test number 4 that I should ask you a question in terms of relative to the EKG, where and why does the first heart sound occur or the second heart sound occur? What do you think? Does that seem like a reasonable question? Mike's checked off on, so it's good to go.
Is it the QRS complex?
Nope. QRS complex is the depolarization of ventricles. T-wave. T-wave is what?
The ventricular repolarization.
That's right. We got to turn off the ventricles. That's our T-wave. That's the repolarization of the ventricles. And so when the ventricles stop contracting, the pressure in the ventricles drops below the pressure within the aorta. And so the pressure in the aorta causes the leaflets of the aortic valve to slap together, causing the second heart sound. Or we could say the pulmonic valve. They're happening both at the same time. abnormal heart sounds are referred to as murmurs. And in thinking about valves, there are two types of valve disorders. Just back up for a moment, Logic 101. Abnormal heart sounds occur because there's damage to the valves themselves. That's what causes the abnormal heart sound. So by definition, we're going to say that if an individual has a murmur, we're saying they have an abnormal heart sound, and depending upon whether it's the first or the second heart sound tells us whether we're talking about aortic valves or, aortic/pulmonic valves or AV valves. And the types of abnormal valve disease are you can have stenosis. That's a narrowing of the valve. And you can have regurgitation. Which is a backflow of blood. So for instance, if you had regurgitation, let's say due to the left AV valve, that's telling you that the leaflets are not forming a nice tight seal so that blood can leak back into the left atrium, and I've mentioned this before, and back into the lungs. All right, that's it. I'm done talking about all this stuff. But that's a good preparation for the lab we're going to do tonight. So let me just get iWorks up and running here, and then we'll get the show on the road. Just take a quick break here for 5 minutes or so. Let me get this set up.